All right, so for this lab, we're gonna be demonstrating genetic drift. So I have an initial population here. So we have a lot of individuals. There's roughly 3,000 of them. And so we have two different alleles. I have allele 1 and allele 2. And so what we're gonna try and demonstrate is how do these random population decreases impact our allele frequency. So I'm gonna take this cup, and what we're gonna do is we're just gonna take a small random sampling, which is roughly half a cup of this population. So what we're saying is these individuals have either died or stayed on the mainland, and these individuals are gonna form our brand new population. So what we're gonna do here is just figure out, okay, we knew that this population had 50% of allele 1 and allele 2. So if we reduce our population size, how does that impact our new allele frequencies? All right, so we took our population size and we separated them out, and you can see here that based upon counting all of them, allele 1 had 190 individuals who had that particular allele, and for allele 2, we had 203. So you will be calculating the allele frequency to figure out what it was for each one of these alleles. And so next what we're gonna do is test a smaller sample size to see if the allele frequency changes more than it did with a slightly larger population size. All right, so this time we're now gonna take a smaller sample size, right? So I'm gonna take a random sample size, but this time I want my overall population to be smaller than our last one, and see, does a smaller population change the allele frequencies more than in that first trial and sample that we did? All right, so we did our quarter cup sample, which is roughly half the size of our last one. And so you can see here when we separated them out and counted them that for allele 1, we had 104 individuals with this particular allele. And for allele B, we had 111. So you're gonna perform the exact same calculations on your data table as you did for the first time. And then you are actually gonna be performing the other trials here. So we looked at both a quarter cup sample, a half cup sample. So now you're gonna take even smaller samples, and you're gonna repeat this exact same process using a population size of 50. And then you're also gonna be repeating it 3 times for a population of 20 to understand if we decrease that population even more, how might that impact the overall allele frequencies.